Hello, I am JL Santos and I will be presenting my research to a lay audience. My research involved the investigation and mutation of Pus7. So Pus7 is a protein called pseudouridine synthase 7, and this protein changes the urine or uridine of RNA to an alanine. This is conserved from yeast to human, and this protein has signals that alert the cell of its environment. In this particular research, I will be giving the stress signal environment of heat and how that affects the protein. Mutations in this protein can cause intellectual disability and growth retardation. This is an overview of the steps during my research. So first, we select a mutation in Pus7, and later on I will explain the methods of doing so. Then we designed a primer. Then next, I mutate the gene in a plasmid. And so in this photo, this is a plasmid map. And so a plasmid is a closed, circular, self-replicating DNA. And this has a gene area, a promoter, restriction sites, and things of that nature. And the fourth step is making lots of copies of the mutated plasmid, and we will do that by putting the plasmid into bacteria. Specifically, we will be putting it into E. coli, where it can multiply and we can have lots of copies of it. Then we will be putting those copies of the plasmid into Baker's yeast. We will be putting it into Baker's yeast because there is no Pus7 in that yeast, and we want to just grow the Pus7 mutation into the yeast. And then next, we will be testing our mutation in a plating assay and see if it can survive the heat stress we will be putting on it. So these are the methods that I used during my research. So first, I selected a mutation on Pus7 using a program called Chimera. And on the right photo, this is a picture of the Pus7 gene structure. And on the left, this is the Chimera program and where I chose to do my mutation. And so I chose to do my mutation in the green area, and then I designed primers for it. In the green area, I was able to find the different DNA sequences, and in doing so, I did the mutation of changing the U to an A in this red, and the black is my primers. And so after that, we did a site-directed mutagenesis, and that is just a method in cell biology where I am able to specifically and intentionally mutate an area of the protein. And then after I was able to do that, I inserted that mutation into a plasmid, into the gene region of the plasmid, and then after that I put it into bacteria, and then I tested it. And so in this next slide, it is my results for the testing. And so what I did is I took my plasmids and I put it on a galactose gel, agarose gel, and in that so in galactose, Pus7 is able to be expressed. That's why I grew it in galactose. But we put it on these gels where I just plated it right on top, which are these little droplets, and I put them into 23 degrees, 30 degrees, and 37 degrees. And PUS7 really likes the 30 degrees. It's really happy there. And 23 is a bit too cold, and then 37 is a bit too hot. And I did the testing in different dilutions, and so that's why there are 4 columns in each or 4 photos in each row. And so the first one is 1 dilution, the second one is 10, and then 100, and then 1,000 dilutions. And so the first column here is the URA. This is just there's no mutation in it, and so as you can see, there was nothing really expressed because there was no mutation there. And then we have the wild type, which is the yeast with the PUS7 on a plasmid. And for this, you can see that the saturation of the image shows that there was a lot of growth in that area, and you can see that there's good growth in these. And then you can see in the 37 it's a bit cloudy. And then we have a control, which is just yeast with a D256A PUS7, which is our control. And then we have a mutant PUS7, or a mutated PUS7 in it. And by my results, you can see that they were able to survive in these conditions. And so that is my conclusion, is that their mutation was successful, and PUS7 was able to be conserved. Or sorry, it was able to be expressed. And this is important because recently in this paper, or in this article that I found, a novel PUS7 mutation causes intellectual disability and autistic and aggressive behaviors. And these were done by these doctors that saw that there was a mutation in PUS7 that caused these behaviors. And so the study and research of PUS7 can be really important for the future, as it can help with altering these autistic and aggressive behaviors in humans. Thank you.